Hello everybody, welcome back. This is Mr. Henson. Welcome back to Molecules to Metabolism. So last lesson we were looking at the structure of amino acids. We said that they always had the same kind of structure. You've got an amine group, which is your NH2 group. You've got your carboxyl group, which is your COOH group, okay, which is your acidic group. So you've got your amine and your acid group, and that's what makes it call it our amino acid. You've always got this NCC backbone. You've often got an R group there, and now these R group are the variable groups, so that can be different. For each different of the 21 amino acids, the R group is different, okay, and that's what makes our 21 amino acids different. They also have a hydrogen coming off the central carbon atom, okay, but essentially they've all got identical structure. All right, well, let's look at a couple of other types of molecules, okay. Let's look at our fatty acids. So fatty acids have, again, it's pretty much a set structure. You've got our carboxylic group, which is the same as in our amine, double bonded oxygen, single bonded hydroxyl group, and then you've got this area up here. Now this HC3 just means a C with 3 hydrogens bonded to it. The fourth bond of the hydrogen links back to repeating units of CH2. Okay, and that's why there's an N there, because these CH2s can go on and on and on and on for a very, very, very long time. All right. Now, if we're looking at our sugars, we're looking at our most simple sugars, or our monosaccharides. Mono meaning one, saccharide meaning sweet. Okay, one sweet or single molecule sugars. A couple of examples are glucose and ribose. Glucose is what we call a hexose sugar. It has 6 carbons in it. Okay. So you've got carbon 1 on the right-hand side, all the way around going clockwise to your last carbon, which is carbon 6. So you always start, when you're counting carbon atoms, on the right-hand side, and you count in a clockwise direction. Ribose is a pentose sugar, meaning, of course, that it's got, yep, you guessed it, 5 carbon atoms. Okay, and again, carbon 1, 2, 3, 4, and number 5. Now, you may have come across ribose before, because, of course, it's the sugar that we find in DNA. So deoxyribose nucleic acid. So it's deoxyribose is a little bit similar, but it's got 1 less oxygen in it than ribose sugar, but otherwise it's absolutely the same. You can see a few other examples down here of other sugars. Okay, you've got glyceraldehyde, ribose, glucose, galactose, and fructose. Okay, so these would all be monosaccharides. All right. Now, all monosaccharides have ring structures in water, so they all form this big ring structure. Now, at the start of this presentation, we talked about the term metabolism. And metabolism of an organism being all the chemical reactions that happen in a cell of an organism. So chemical reactions in organisms come in 2 different types. We have ones that are called anabolic, which means we're building up complex molecules from simpler molecules. And we have catabolic reactions, or catabolism, which is the breakdown of complex molecules into simpler molecules. Okay, so a couple of important terms to remember: that condensation reactions make bonds and release and are water releasing, and these are our anabolic reactions. So it's the building of molecules. Okay, so for instance, protein synthesis is an anabolic reaction, the joining together of amino acids to make polysaccharides or proteins. Okay, the hydrolysis reactions, they break down bonds, and it's hydrolysis because it's water splitting. So these are catabolic reactions. And these are the ones which break down molecules. So, for instance, digestion, chemical digestion involving enzymes, is the breakdown of complex molecules into smaller, simpler ones. Okay. Now, it's important to remember that all of the reactions in these different things require enzymes or these biological catalysts in order to make them work. So, let's have a look at our condensation reactions here. All right, you've got 2 monomers. Let's say you've got two monomers, single molecules. Okay, maybe it's glucose, maybe it's pentose sugar. So what happens is there's a reaction involving an enzyme synthase. So it's synthase because it's an enzyme which is going to be synthesizing something else. And at the end, we're going to be joined together. All right, and they're going to be bonded in them here. All right. The opposite to that, of course, is the hydrolysis, where they're being split apart. So we have a hydrolase, or an enzyme which is breaking down this bond here, and so we've got two monomers out at the end. All right. So remember, in synthase reactions or in those anabolic reactions, water is released or water is given out, and in our hydrolysis reactions, water goes in. Okay. So an example of a condensation reaction, maltose synthase condenses two molecules of glucose into maltose, forming what we call a glycosidic bond. So you can see here you've got glucose, and over here you've got another glucose, and you can see these two hydroxyl groups here, look. Okay, now during this reaction, you can see it now. All right, this glycosidic bond is forming, okay, and water is coming out of the reaction. So water is a byproduct. So now they're sharing this one oxygen of these two, and water is a byproduct. Okay. A ribosome condenses two amino acids into a dipeptide, forming a peptide bond. Okay, so here's amino acid 1, amino acid 2. Okay, a condensation reaction occurs, okay, in the carboxyl group, okay, and between the amine group of another amino acid. And so what happens again, okay, is the H2O is released as water, okay, and a bond forms between the carbon of one carboxyl group and the N of the amine group of the other amino acid. Okay, the bonds formed are a type of covalent bond because they're forming between non-metals, and so that's those types of reactions. So examples of hydrolysis reactions. Let's think. Okay, you can see here amino acids that have been joined together. So a protease hydrolyzes a dipeptide, meaning two, into two amino acids by breaking the peptide bond. So where it goes NCC, you can see the carboxyl group here is joined to the amine group of the next amino acid. Okay, what's going to happen here, look, is H2O is going to be used, it's going into the reaction, and it's creating this amino acid here, and this amino acid on this side, okay, and so water is actually going into using that reaction. Lactase hydrolyzes lactose into glucose and galactose, okay, breaking the glycosidic bond. So here's lactose, again, which is a disaccharide, meaning two sugars, 1, 2. Okay, lactase and water go into the reaction, okay, and that glycosidic bond that's there, look, is being split apart, okay, and obviously water has to be added into that reaction so that you've got your two hydroxyl groups that remain on your two molecules that are left over. So you might see the term dehydration reaction mentioned interchangeably with condensation reactions. Technically, a dehydration reaction is when the water molecule has come from one of the reactants, whereas for a condensation reaction, part of each water molecule has come from each reactant. So in the case of the previous slide, hydroxide from one glucose and the hydrogen from the other. Okay, just something to be aware of. Okay, this is our last slide, so hopefully we should be finishing by 10 minutes. Okay, so it was originally thought with these biological molecules that there was this theory called vitalism. And vitalism basically meant that things, organic molecules, could only be created with a vital principle, i.e. by a living organism. So that things like glucose being changed into, glycogen, which is a polysaccharide, okay, you needed that vital principle to make that happen, okay, or a living, something that was living to do it for it. Now, Wohler, who was a German scientist, accidentally synthesized something called urea in 1928 whilst attempting to prepare something called ammonium cyanate. So in a letter to a colleague, he says, "I can no longer, so to speak, hold my chemical water and must tell you that I can make urea without needing a kidney, whether of man or dog. Okay, now this idea really undermined the vital principle and was part of the evidence which then went to falsifying the theory of vitalism, which said that organic chemicals could only be synthesized by living organisms. So vitalism nowadays has no credit as a theory, but the above statement is seen by many from a historical perspective as to be untrue. For an outline on vitalism, you can read this article here. And the application statement above implies that the central tenet of vitalism is only organisms can synthesize organic compounds. This was not accurate. In essence, vitalism proposes that an unknown factor is essential in explaining life. And vitalism on this premise is both unscientific and unfalsifiable. Okay, thank you very much for listening, and see you next time.